All right, so let's look at nucleic acids. We're going to make a graphic organizer of this. It's going to be split into about 3 columns. The first one can be a little bit, it's like the size of a piece of paper. First one can be a little bit skinnier. And then we'll have 2 columns over here. You can leave the space on the right, all right, like without drawing it, but just to kind of give you an idea. All right, so nucleic acids. Nucleic gives you a clue that these have to do with the nucleus, although, and that's DNA is true, RNA is outside the nucleus, but as well as inside the nucleus, but you know, these are nuclear related. So let's think about the common elements in nucleic acids, especially the unique ones. So we've got our common CHON, carbon, hydrogen, oxygen, nitrogen, and then there's one that is, it's not unique, but it's more unique, the phosphorus. Phosphorus is also in phospholipids, which are not nucleic acids, but it's not in proteins, it's not in carbohydrates. So phosphorus can be used to label nucleic acids. If we track the phosphorus, sometimes we can track it instead of the proteins. So what are identifying features or identifying functional groups? FXNL is functional groups. The phosphate group. And remember that has a negative charge, which makes both DNA and RNA, which are the nucleic acids, negatively charged. You can do this phosphate group. All right, what's the monomer of nucleic acids? The single subunit that builds a nucleic acid is the nucleotide. And it's technically the triphosphate form where it has 3 phosphates on it, so it's high energy. ATP is a nucleotide for RNA. It's also the energy molecule of the cell. And we'll draw that in a little bit. What's the polymer of nucleic acids, which is the long string of nucleotides? We can call that a poly, meaning many, nucleotide. That makes sense. And when the nucleotides are stuck together into a polynucleotide, they all just have 1 phosphate because they lose the 2 when they're bonded together. So they're in their monophosphate form. That's not the most important thing. Nucleotide and polynucleotide are much more important than the monotriphosphate, but I want to be thorough with this. So what are the types and functions of nucleic acids? We've got DNA. DNA's function is to store information. And what's that information for? To make proteins and RNAs, functional RNAs. So RNAs with a job. We'll also note that DNA is hereditary, which means you can pass on traits. That's how you pass on traits or alleles, different versions of genes. The second type of nucleic acid is RNA ribonucleic acid, which we'll detail in a little bit. It's highly varied in structure and function. Here's just some examples. So different types of RNA can transfer, like mRNA, the DNA information to the ribosomes to make the protein. That'd be mRNA. We also have RNAs that control gene expression. Basically, they can help turn off or on genes, like microRNAs, siRNAs. There are other RNA types as well, which we'll learn about throughout the semester. So let's look at the monomer. We'll just remember that. So it has 3 parts to it. So we'll draw 3 phosphate groups. I'm going to draw a wiggly bond in between them, because they're high energy. Then out to a little arm. That's a carbon on the end of that, or the bend of it. And we have a pentagon with an oxygen in it. That's the ribose. And then we have a group here, which I'll detail a little bit more, but we're going to label it NB, the nitrogenous base. So let's number these. 1, I not have room. 2, 3, and on this pentagon, let's draw an OH group. And these have polarity, so let's label 1 and 3 prime, which looks like a 3 with an apostrophe, but it's like straighter than apostrophe, and 5 prime, the phosphate end. So what are these groups? Number 1 is the phosphate group. Number 2 is that ribose or deoxyribose, so we'll call it a pentose, meaning 5 carbon sugar. And the carbons are 1, 2, 3, 4, 5. That's why this is called 3 prime here. That's the 3rd carbon in the ring. And this is why the phosphate is called 5 prime side, because that's the 5th carbon in the ring. And the NB part, number 3, is the nitrogenous, nitrogen-containing base. If we look at just the pentose sugar and the nitrogenous base, call that a nucleoside. And then once you add the phosphate groups on, that's the nucleotide. Nucleotide is the monomer. So let's look at these groups in a little more detail. Draw a line. This will help us distinguish RNA and DNA. So we have the phosphate group. If you have room to the right, you can draw it there, or you can draw it below. Okay, So important thing here is that this is a negative charge. We draw this group in a circle a lot of times. You see we draw the P in a circle. So we can kind of do that here. The carbon is the 5' carbon on the corner, and then the rest of the molecule. But that's not part of the phosphate group. That's just where it's attached. So this means DNA and RNA are both negatively charged. Let's look at the pentose sugar. So that means 5 carbons. As I mentioned before, you label them 1' through 5'. So it said 1'. 2', 3', 4', 5'. So let's draw those out. So the oxygen, and then pentagon with this arm up here. One prime carbon, 2' carbon, 3' carbon, 4' carbon, 5' carbon. The 5' carbon is attached to the phosphate group, and 3' carbon is attached to the OH, the hydroxyl group. That's important because that's where the reaction is. The reaction to build polymers happens at the hydroxyl group and the phosphate group on the next one, really. The phosphate group on the next nucleotide attaches onto the OH group of the previous nucleotide. Then we have our nitrogenous base. You can also just call it a base. Nitrogenous comes from the fact that it has nitrogens in the ring. And these are flat shape, which is important because that structure allows DNA and RNA to have different functions or be able to interact with planar molecules like flat molecules. There are two categories of nitrogenous bases. There are purine bases. And then way over here, I'm going to write pyrimidine bases. Purines, you don't need to know this exact structure, but they're 2 rings. All right, we have 2 types of purines. We have adenine, that's a nitrogenous base, adenine, and we have guanine. Adenine we abbreviate A, guanine we abbreviate G. So if you've ever seen ATCG, that's what these guys are. They're the nitrogenous bases. Pyrimidines have a single ring, And those guys are 3 different bases: thymine, T, cytosine, C, and uracil, U. It's all capital letters, so I'll draw it like that. So one way you can remember which ones are pyrimidines, which ones are cysteines, well, at least for thymine and cytosine, they have the Y for pyrimidine in them. I remember pyrimidine because it's like pyramid. So if you remember from geometry, or you can think of the ancient Egyptian pyramids, they have they're like this. So pyramid is on one base, or a single base, which is like a single ring. That's how I remember it. And uracil, you'll learn that uracil and thymine are the versions that are in DNA and RNA, just the difference there. And so the uracil and thymine are both pyrimidines. Let's just draw a little nitrogenous base here so you can see the nitrogens. You definitely don't need another structure at this, like exactly at all, but it's useful to see, I think. Let's do it like this. Okay, so I'm going to draw nitrogen here, straight, straight. So this is the pentagon part. And then I've got a hexagon, the nitrogen in the ring, another nitrogen in the ring. Yeah, that's technically 6-sided. Not the greatest, but got some double bonds in there to make the electrons shared. And so then you can see those nitrogens in the ring there. That's the nitrogenous base. Okay. You can pause that, of course, if you need to. And then we have our 2 types of nucleic acids, DNA and RNA. We'll compare them side by side. Let's look at the names. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So where do these words come from? The deoxyribonucleic acid is where DNA comes from, and ribonucleic acid is RNA. Deoxy is referring to without oxygen. So this one has 1 less oxygen than the ribonucleic acid. So let's look at that. This is point number 1. Point number 2, just so we can match them up. There's our carbon here. And actually draw it in the ring this time. It's our pentose sugar. We're going to look at the difference between those. This is the difference right here. So both of them have the OH here. But the difference is this guy without oxygen. It's just a hydrogen, no oxygen there. So this type of pentose sugar is called deoxyribose. Versus pentagon here, OH, but we have another OH there. So that's the one with the oxygen. And not surprisingly, this is called ribose. Point number 3, to compare them. A matches with T, so they hydrogen bond together. And we'll see the structure where this happens in a minute. So they have 2 hydrogen bonds. C hydrogen bonds with G. It has 3 actually bonds. Okay. What about in RNA? A, this is different, is with U, uracil. Again, those are hydrogen bonds. And C, again, is with G. These are not the same C and G, though, right? Because they have a different sugar. I mean, the C and G are the same, but the sugar is different in the molecule. But hydrogen bonds, hydrogen bonds, hydrogen bonds. But it is the same cytosine and guanine, actual nitrogenous base. Point number 4, DNA is double-stranded. And RNA is single-stranded. You can draw that here. This is not the greatest picture. So there's your double strand. It's held together by these hydrogen bonds between those nitrogenous bases that we just saw. We'll see that more detail in a different chapter. We've got a phosphate on one end here, and if we track this down, this strand ends up on this left side. So we have the hydroxyl group over here, 5', 3'. Let's do this in a different color. Okay, and then the other one, we have it the opposite with the phosphate here. We track it up. Good, it's on this side. So the hydroxyl group's over there. So this is what we call anti-parallel. Saw that in the book. Anti-parallel means that 3' and 5' are on opposite ends from each strand. That's just how the DNA fits together. One strand is oriented 5' to 3'. And the other, looking from the same end, is 3' to 5'. If we unraveled it, it would be like this. There's the 2 strands. That's your untwisted DNA. So it'd be better to color those. DNA is anti-parallel, and that is important, as you shall see, in the genetics unit. So we've got single-stranded RNA, 5' phosphate on one end. Then we have the nucleotides, the ribonucleotides. And then we have the OH on the other end. So it also has polarity to it. You may see this folded on itself. Something like this. We've got the phosphate on one end, the hydroxyl group on the other end, and then this has hydrogen bonds. It's still just a single strand. Hydrogen bonds can hold it together. in three-dimensional shapes. Again, 3' 5'. Okay, so this is like an example of a shape. And that means that RNA has many functions because it can be many shapes. Or it might not be folded at all. Those are the basics of what I have to tell you about nucleic acids.